Dr. Anjali Venkateshwaran, and we'll be introducing today's speaker on behalf of Biomir, a preclinical CRO with locations in Boston, San Francisco, and China. Welcome to the webinar series on novel approaches to cell and gene therapies. The speaker for today's webinar is Dr. Miguel Munoz-Alia. Dr. Munoz-Alia is a research associate in the Department of Molecular Medicine at the Mayo Clinic. He received his PhD in cell biology and genetics at Autonomous University of Madrid, Spain, after completing his bachelor's degree in biology. He began his career at the Spanish National Reference Laboratory for Measles and the Spanish National Center for Biotechnology, where he acquired a keen interest in molecular epidemiology and evolution of measles virus genotypes. In 2015, he joined the Mayo Clinic, where he is working on the clinical development of oncolytic measles viruses and measles virus vaccine vectors. The title of Miguel's talk is Retargeting and Shielding Measles Virus Vectors for Oncolytic Virotherapy. And without further delay, it is my pleasure to introduce Dr. Miguel Munoz-Alia.
Thank you, Angie. It is my pleasure. Thanks for the kind introduction. Hello, everyone, and thank you very much for taking part of this presentation where I'm going to speak about retargeting and shielding of recombinant measles viruses for oncolytic virotherapy. Before we get started, I need to disclose that the work I am presenting has been subjected to a patent application which has been sub-licensed. Therefore, there is a financial conflict of interest related to this research. Oncolytic viruses are an emerging class of anti-cancer weapons. One of the key features of viruses is their natural propensity to infect malignant cells because of many incompatibilities between the angiogenesis process and the maintenance of an antiviral state. When the virus infects the tumor cells, it auto-amplifies and replication eventually causes cancer cells to burst, killing cancer cells and releasing tumor antigens. These tumor antigens next trigger a cellular anti-tumor immune response that can not only eliminate any remaining tumor cells nearby, but can also reach distant and uninfected tumor cells, resulting in tumor clearance. There is, however, an overarching question. How do we deliver the oncolytic virus? We can get the oncolytic virus to the tumor via intratumoral or intravenous delivery. In the intratumoral delivery, the virus is injected into a single lesion or region. In red here, we have an infectious center that is seeded into the tumor in blue. After a certain time, infected cells at the core of the infectious center begins to die in black, leaving a ring of viable and infected cells. As infection continues to spread, the infectious centers continue to enlarge, but the ring of viable cells is maintained until the infection reaches its maximum spread and all infected cells die. This IT delivery works for some oncolytic viruses, such as TVEC, which is the first ever oncolytic virus approved for the treatment of patients with melanoma. However, as illustrated in the graph, intratumoral dispersion might be poor and tumor regression might be mainly mediated by the immune system rather than direct virus oncolysis. A good option for the IT delivery could be intracavitary delivery. On the other hand, in the intravenous delivery, multiple infection centers are seeded and they eventually coalesce to form large regions of infection. This IV delivery is potentially appealing to tackle metastatic lesions. However, there are multiple obstacles to the IV delivery, such as dilution of the virus in the blood, neutralization by antibodies and complement proteins, and sequestration of the virus by hepatic or splenic phagocyte. If the virus circumvents these obstacles, the virus still needs to extravasate from tumor blood vessels and will need to diffuse throughout the extracellular matrix. All this being said, there are potential solutions to each of these obstacles. However, in this presentation, I will mainly focus on the problem of neutralization by antibodies for a particular oncolytic virus, which is the measles virus. The principle of using measles virus against cancer arose from the observation of a young boy in Africa who was reported to have an aggressive lymphoma, but developed childhood measles, and he had a dramatic remission of his cancer, as you can see here in the picture. This fact made scientists think that perhaps viruses could have an effect on killing cancer cells. So flash forward many years, here in the Mayo Clinic, a measles virus vaccine strain was designed with the goal of that the virus will attack cancer cells preferentially about normal tissues and kill them. Of the different clinical trials here at Mayo Clinic using measles virus, you might have probably heard about the myeloma clinical trial, since it attracted a big deal of attention worldwide. In such a phase 1 dose escalation myeloma trial at Mayo Clinic, one of the patients that received the highest dose of the measles virus, actually 1 million times more than the dose we received in the actual vaccination for measles, had a fever over 105 degrees Fahrenheit during her infusion, and also had a severe headache that lasted for several days. She, however, saw a drop in her serum free light chain and resolution of all myeloma cells in the bone marrow. Also, a number of plasmacytoma or lumps of myeloma cells growing from her bones, some of which were actually even visible, especially one growing out of her forehead, shrank. And they shrank visibly to the naked eye and also by PET scan imaging. One of the characteristics of this patient was that she was seronegative for measles virus. Moving forward, patients will be asked to have low titers of antibodies for enrollment in subsequent phase 2 trial. However, since we all have been vaccinated against measles, we will all have antibody titers against the virus, and it will be difficult to recruit a sufficient number of patients. Therefore, measles virus antibodies can be considered a frenemy, because neutralizing antibodies provide that protection from measles after vaccination or natural infection, but they also limit the therapeutic effect of oncolytic measles virus. Here we can see that if we partially immunize CD46 transgenic mice with human antisera prior to the infection with a measles virus expressing luciferase, we can see a decrease in luciferase expression in those mice that were immunized versus those that were not. And this inhibition asserted by neutralizing antibodies is dose dependent, reaching a statistical significance when we use a concentration equal to 6 international units per milliliter. Recently, this dual role of measles virus antibodies have been used as an argument to oppose measles vaccination. Anti-vaxxers now claim that they do not want to get vaccinated against measles because if they develop cancer later on, they will not be eligible for treatment with oncolytic measles viruses. This is the reason why we published this special article, so as to address some of the misinformation that was being used. In addition, in this presentation, I'm going to show you the new virus that we can potentially use to treat cancer in the case of measles seropositive individuals. But what is measles virus? Measles virus is an RNA virus with a lipidic envelope that enters the host cell via cell-to-cell fusion at the plasma membrane. The entry process of the virus is mediated by the two envelope embedded proteins named the hemagglutinin protein or attachment protein and the fusion protein. The hemagglutinin protein is involved in the first step in virus entry, which is binding to host cell receptors. There are 3 known receptors for the measles virus, 2 of which, SLAM in immune cells and Nectin-4 in epithelial cells, determine the pathogenicity of measles virus isolate. CD46, on the other hand, can only be used for attenuated and vaccine strains of the measles virus. Regardless of the receptor use, binding of the hemagglutinin to any of the receptors activate the fusion. Again, okay. I'm gonna start again with this slide. But what is measles virus? The measles virus is an RNA virus with a lipidic envelope that enters the host cell via cell-to-cell fusion at the plasma membrane. The entry process of the virus is mediated by the two envelope-embedded proteins named the hemagglutinin protein and the fusion protein. The hemagglutinin protein is involved in the first step in virus entry, which is binding to host cell receptors. There are 3 known receptors for the measles virus, 2 of which, named SLAM in immune cells and Nectin-4 in epithelial cells, determine the pathogenicity of measles virus isolates. CD46, on the other hand, can only be used for attenuated and vaccine strain of the virus. Regardless of the receptor used, binding of the hemagglutinin protein to any of their receptors activate the fusion protein to trigger membrane fusion. Because a video is worth 1,000 words, here in this video, I included this video about the virus fusion entry process. On the left-hand side, we have the hemagglutinin protein, whereas on the right-hand side, we have the fusion protein. We have just seen that the hemagglutinin protein interacted with the host cell receptor, and this interaction activated the protein to undergo a set of conformational rearrangements, which are going to subsequently trigger the fusion protein. We do not know how this F protein receives the signal from the H protein and how H coupled receptor binding with F triggering. Nevertheless, the fusion protein senses this interaction of H with the receptor and undergoes extensive conformational rearrangement, transitioning from the prefusion conformation to a post-fusion conformation, where the 2 membranes are pressed together and the 2 outlet leftlets fuse. Looking under the microscope, here we have a time-lapse images of viral cells infected with a GFP-expressing measles virus. That's why we start seeing GFP-positive cells that start fusing to each other, growing in size until the whole monolayer is a giant syncytia. As I mentioned before, interaction of the measles virus with any of the known receptors will trigger cell-to-cell fusion. However, it is the frequent overexpression of CD46 in a number of malignant tumors what has prompted the clinical translation of measles vaccine strains as an oncolytic virotherapy platform. However, again, measles vaccination or natural infection induce the production of measles virus neutralizing antibodies that prevent the virus from entering the cell via targeting the 2 surface protein. To solve this issue, we here have replaced the membrane fusion machinery of the measles virus by the homologous complex from the closely related member of the paramyxoviridae family, canine distemper virus. As I'm going to explain shortly, this new envelope, oh man, I'm gonna start again. I don't know why it keeps moving sometimes. I think it's the pointer. As I mentioned before, interaction of the measles virus with any of the known 3 receptors will trigger cell-to-cell fusion. However, it is the frequent overexpression of CD46 in a number of malignant tumors what has prompted the clinical translation of vaccine strains as an oncolytic virotherapy platform. However, again, measles vaccination or natural infection induce the production of measles virus neutralizing antibodies that prevent the virus from entering the cells via targeting the 2 viral surface proteins. To solve this issue, we have replaced the membrane fusion machinery of the measles virus by the homologous complex from the closely related member of the paramyxoviridae family, canine distemper virus. As I'm going to explain shortly, this new envelope was next retargeted towards CD46 via single-chain antibody fragments, so the virus could only infect and produce fusion exclusively in CD46 expressing cells while avoiding anti-measles virus immunity. CD46 canine distemper virus was chosen. Canine distemper virus was chosen among all the other members of the paramyxoviridae family because of his strategic location in the phylogenetic tree. It is said that for 2 viruses to be antigenically different, there must exist at least 20% divergence at the amino acid level. When accounting for this amino acid divergence in the attachment protein, rinderpest virus could have been used in theory as an alternative heme-agglutinin protein because of a greater than 40% divergence at the amino acid level with the measles virus. However, the fusion protein is much more conserved in the family, and we needed to go farther down the tree to get a fusion protein with a greater than 30% divergence at the amino acid level. Then canine distemper virus, or CDV, was the closest virus that we could have probably used. Here, I also want to bring your attention to Nipah virus because I will be showing some data in a few slides. In any case, when we talk about canine distemper virus, we are talking about at least 19 different genetic groups, and the selection of the genetic group could have implications in our success because of host tropism. What I mean to say is that viruses belonging to the America1 genetic group can use jet to identify receptors. For this reason, we focus on two wild-type isolates, 5804 and SPA Madrid. We can see here that the Understeport vaccine strain, which belonged to America1 genetic group, not only infected viral cells expressing canine SLAM, but also the parental viral cells. By using recombinant viruses expressing the green fluorescent protein, we can more clearly observe that the measles virus vaccine strain infect CHO cells when they express CD46, Nectin-4, or SLAM. On the other hand, the Understeport could infect even the parental CHO cell line expressing no known receptor. Since we cannot detarget tropism from a receptor that we don't even know, we focus on the two CDB isolates that I mentioned before, 5804 and SPA Madrid. How did we do that? So here we have a schematic representation of the heme-agglutinin protein, which is a single pass transmembrane protein type 2 that forms tetramer. Its subunit consists of a globular head, a stalk domain, and a cytoplasmic tail preceded by a transmembrane region. To first see if we could expand the tropism of the protein, we display on the C-terminus domain of the protein a single chain antibody fragment, and we chose initially CD38 fused to a histag as a target, as we have previously reported for the measles virus. When we transfected this construct into mammalian cells and performed Western blot analysis, we could see that the single chain antibody fragment was successfully displayed on the protein, as indicated by a delay in the electrophoretic mobility. Here we have one of the original CDBH proteins, and here we have the same one with the single chain antibody fragment attached to it. For comparison purpose, we also display the single chain antibody fragment onto the attachment protein of the Nipah virus. Because it has been shown because it has successfully retargeted and is currently being used in vivo targeting therapies. Flow cytometry analysis saw a comparable level of surface and total expression for the attachment proteins that we use throughout our study. Measles virus H, Nipah G, and CDBH isolate SPA and 5804. Next, since the proteins were similarly expressed on the cell surface, we look for the one giving the highest fusion phenotype. And for this, we use the dual split protein quantitative fusion SI. This fusion SI works by co-transfection of effector cells with one dual split protein plasmid plus the fusion machinery of your preferred virus. In parallel, the target cells expressing a receptor are transfected with the other dual split protein plasmid. When the two cells, one second, someone is calling me and is annoying me. I don't have my phone here, but I have the Apple Watch and it's vibrating.
I did not hear that come through.
Yeah, because I mean, it was my Apple Watch. It was like vibrating all the time. Like,
Just distracting you.
Yeah, it was distracting me like, oh my gosh. and they
No
keep
problem.
falling.
I'm going to edit out all this stuff. So when you're ready, just jump right back in, okay?
All right. Next, since the proteins were similarly expressed on the cell surface, we look for the one giving the highest fusion phenotype. And for this, we use the dual split protein quantitative fusion SI. This fusion SI works by co-transfection effector cells with one dual split protein plasmid plus the fusion machinery of your preferred virus, here denoted as H and F. In parallel, target cells expressing the receptors are transfected with the other dual split protein plasmid. When the two cells are co-cultured, if there is interaction between the fusion machinery in the effector cell and the receptor in the target cell, there will be mixing content, reconstituting the otherwise inactive luciferase protein, whose activity cannot be used as a proxy for fusion. When we perform this SI with the different combinations of the attachment and fusion proteins, we can see that in CHO cells expressing either the His pseudoreceptor or CD38, the homotypic combination of CDB, H, and F proteins from the isolates ESPA or 5804 produce comparable levels of fusion, and they surpass that obtained by the NIPA fusion machinery. Surprisingly, we observed that the heterologous combination comprising the CDB-H 5804 and CDB-F ESPA resulted into a statistical significant increase in the luminescent signal, both in CHO anti-His cells and CHO CD38 cells. And moving forward, this CDB-HF combination was chosen for further modifications. However, we first wanted to study why the heterotypic combination was hyperfusogenic in relation to the homotypic one. And for this, we postulated that the enhanced fusion activity could be related with a lower binding avidity at the HF interface. This assumption was based on the observation that the HF dissociation is essential for the fusion process, as you might remember from the video I showed you before in the beginning. So in order to test our hypothesis, we perform co-immunoprecipitation analysis. In the schematic, we can see that if the molecule magenta interacts with the molecules in green and in orange, providing that we immunoprecipitate the molecule in magenta, we will detect by Western blot analysis the molecules in green and in orange. So what we did was to co-transfect cells with the CDB-F ESPA and either the homotypic or the heterotypic H combination. We next detected the CDB-H in the input versus in the immunoprecipitative fraction after incubation with an anti-CDB-F antibody. We can see that the amount of CDB-H was comparable for both H-ESPA or H5804. However, when we immunoprecipitated F, we barely detect H5804, and therefore the IP input ratio is lower than 1. This means that the HF interaction is weaker for the heterotypic HF pair, and that this weaker interaction translates into an increased fusion activity. Next, we needed to detarget the tropism of the new envelope because, as we can see here, using BSB pseudotype with the CDB-HF envelope and expressing the green fluorescent protein, the virus with this new envelope can infect cells expressing not only DOC-Nectin-4 and DOC-SLAM, but also human Nectin-4 cells. So the targeting from human Nectin-4 cells was achieved via a thyroxine to alanine substitution. And after introducing this amino acid change, we compare the fusion phenotype once again with a fully retargeted Measles virus HF complex. You can see that both Measles virus HF and CDB-HF complexes could only fuse cells expressing CD38 receptor when they display the single chain antibody fragment against CD38, and otherwise they did not produce fusion in this cell line. So now that we have shown that we can retarget and detarget the new envelope, we aim at retargeting the CDB-HF complex towards CD46. And for this, we first produce single-chain antibody fragments against CD46 and assess their binding to CD46 by ELISA. In a third step, these single-chain antibody fragments were displayed onto the H domain of the CDB-H protein to assess cell-targeted fusion. The ELISA results showed that the scFvs bound to CD46, but they did not bind to Nectin-4, which was used as an irrelevant protein. When we display the single-chain antibody fragments on top of the CDB-H, we can see that KO1, which saw one of the lowest binding to CD46, did not drive fusion into CD46 cells. However, the one called AO9, which saw one of the highest binding to CD46, allow CDB-H to produce CD46-dependent fusion with a kinetic comparable to that achieved by the measles virus hemagglutinin protein. So we chose this CDB-H scFv AO9 together with the CDB-F protein to replace the homologous envelope glycoproteins of the measles virus present in the Moraten vaccine backbone, generating a virus that we call stealth. By blotting the same amount of PFUs, we observed using an antibody against the measles virus nucleocapsid that there was a similar particle to PFU ratio. An antibody against the measles virus hemagglutinin protein could only detect the hemagglutinin protein in the case of measles virus, but not in the case of stealth. And the opposite was true when we use an anti-His antibody, where now we can observe a higher molecular weight and we can only detect the hemagglutinin in the case of stealth, but not in the case of measles virus. Using an antibody against the cytoplasmic tail of F, we could also distinguish the two different F proteins due to the different molecular size. We next perform neutralization assays with pooled human sera, and for those that are unfamiliar with the neutralization assays, in this assay we pre-incubate the virus with serum samples before inoculation into cells. This way, the virus can only grow if there are no neutralizing antibodies in the serum samples. Otherwise, the virus will lose infectivity. Since the virus is expressing the green fluorescent protein, we can easily count syncytia formation and plot the infectivity over a range of dilutions. When we do this, we can see a decrease in the infectivity of the measles virus with decreasing dilutions of the human serum samples. In the case of stealth, we do not see this dose-dependent inhibition. We observed the opposite pattern when we use ferret anti-CDV serum. Now, stealth saw a dose-dependent inhibition, whereas measles virus did not. Finally, we wanted to confirm the expected tropism. We can see again that measles virus infected cells expressing CD46, Nectin-4, and SLAM, whereas stealth could only infect those cells expressing CD46. Next, we tested in vivo the utility of stealth as an oncolytic vector, and for this, we implanted into SCID mice subcutaneous U266 myeloma cells, and when tumor reached 500 millimeters cubic, we treated the animals with one intravenous dose of 1 E5 TCID50, which is known to be a suboptimal dose. As we can see in the upper right corner, tumors grew rapidly in the PBS treated control group, and all mice had to be sacrificed by day 12. However, those mice treated with either measles virus or stealth saw a slower tumor progression. We confirmed that the delay in the tumor growth was caused by measles virus trafficking and replication into the tumor, since the virus was administered intravenously and the tumors were implanted subcutaneously. Here we are looking at the IHC staining of tumors of 2 mice per group, where we observe extensive positive staining only when animals receive virus intravenously. Overall, virus treatment resulted into a increased survival of animals, doubling the median survival, and we did not observe a significant difference between measles virus and stealth. We next tested the therapeutic effect of stealth in a more relevant clinical model, focusing on ovarian cancer. So ovarian cancer saw the highest mortality rate of all gynecologic malignancies, with 14,000 deaths per year in the United States. And 70% of the patients relapse after initial treatment that includes surgical cytoreduction and chemotherapy. Also, 80% of patients still have disease confined to the peritoneal cavity at relapse. So ovarian cancer is an excellent candidate for local regional delivery of therapeutics, including viral therapeutics. So in the lab, we established an orthotopic mice model of ovarian cancer by implanting scope cells intraperitoneally into anemic nude mice, which then receive either PBS or measles immune human serum prior to a single intraperitoneal injection of the virus. Animals that were left untreated developed ascites and saw a median survival time of 18 days. Here I'm showing a normal mice versus one with ascites, where the intraperitoneal fluid is quite evident. When mice were treated with a single injection of the measles virus, in the absence of anti-measles virus antibodies, the animals saw a significantly prolonged survival with a median survival time of 37 days. However, when animals were passively immunized, the therapeutic effect was lost. In the case of stealth-treated animals, they saw a significant prolonged survival with a median survival time of 53 days. Unlike measles virus now, when animals were passively immunized, they still benefit from the treatment with stealth, and there was not a statistical significance between the two conditions, meaning antibodies or no antibodies prior to the injection of stealth, whereas there was a difference in the case of the parental measles virus. This experiment was also repeated with scope cells expressing luciferase so as to determine the tumor burden through bioluminescent imaging. At day 0, tumor burden was comparable across groups, as we can see here in the quantification of the signal. But as early as 7 days later, we could see a significant reduction in the tumor burden when mice were treated with either stealth or measles virus in the absence of measles virus antibodies, as we can see here. We do see a significant difference in tumor burden in the animals treated with measles virus, depending on whether animals were passively immunized or not, as we can see here. On the contrary, passive immunization did not result into a statistically significant increase in tumor burden when animals were treated with stealth, although there is a trend towards an increased tumor burden when they were passively immunized. To conclude, I have showed you that the wild-type CDB fusion apparatus can be engineered to efficiently trigger fusion in a receptor-specific manner. We have seen that the targeting from human Nectin-4 expressing cells can be achieved via a substitution in the position 539 in the hemagglutinin protein. And we can retarget the CDBH protein to cancer-selective receptors via display of ligands. We have generated measles virus stealth by replacing the measles virus hemagglutinin and fusion proteins with a CD46 targeted homologous glycoprotein from a wild-type canine distemper virus. Finally, measles virus stealth efficiently targets and lyses CD46 expressing cancer cells in mouse models of ovarian cancer and myeloma, and evades inhibition by human measles immune serum. To conclude, I just want to say thanks to Dr. Stephen Russell, the PI of the lab, as well as Liang Wen, San, Rebecca Nase, and Eugene Ba, because they helped setting up some experiment, and also different collaborators that provided very valuable reagents to perform the work I have just discussed with you. So thank you so much for listening to this presentation. Thank you.
Thank you, Miguel, for a really fantastic and interesting presentation. So I had a question for you. So you'd shown some really interesting data showing the oncolytic effect of the measles stealth virus. Are there plans to do more work looking at the immune response activation using stealth to have a more durable response in mouse models?
Yes, that's a great question. One of the problems trying to test measles virus is that mice are not permissive to measles virus replication.
Oh.
So that's why we need to use either skid mice or nude mice. So as I mentioned at the beginning, so there are two phases. We have the oncolytic phase, and then we have the immunotherapy phase. Interestingly, so the patient that saw the dramatic response to measles virus virotherapy, she did not have neutralizing antibodies against the measles virus, but she had T-cell responses against the measles virus. And we think that that's the case because she underwent like 2 bone marrow transplants. So they might have wiped out the antibodies, but the T cells were still there. So what we believe is that by using this stealth, so we have shown that there are not cross-reactive neutralizing antibodies, but based on the literature, there are cross-reactive T cell epitopes. So we think that now if you are vaccinated against measles virus and we use a stealth, the virus is not going to be neutralized, so it's going to be able to infect cancer cells. And then the persistent anti-measles virus T cells that you may have might come into place and wipe out the rest of the cancers. Yeah. The problem is that the only way to do this in vivo would be by doing a passive transfer of measles-specific T cells. Otherwise, it's kind of very difficult to show. Yeah, but very great question. Yeah.
So I do know that for influenza virus, for example, the ferret model is very, very well studied and is a great model to study infectivity of influenza. I know we're talking about measles now, but are there other non-mouse models like ferret or any other large animal models that can be used to study a more longitudinal response to measles virus, either the wild type or stealth?
Yeah. Well, the problem is, for example, in this case, even if you look into NHP monkeys, there is a key difference between monkeys and humans. And in monkeys, their erythrocytes express CD46, and that doesn't happen in humans.
Okay.
So, now we have this stealth virus that can only use CD46, and probably in monkeys could cause, lymphopenia. Or, but that wouldn't happen in humans because we don't have CD46 in erythrocytes. Right. Okay. So there are
tough to get a model.
It is tough, and this single chain antibody fragment that we use against CD46 is really specific for human CD46, and this virus cannot infect viral cells, cannot use the CD46 expressed on the viral cells. So yeah, it's kind of tough because in ferret, I mean, it's a great model. We could, for pathogenesis, but they are quite aggressive, and the CD46 is quite different, and the only way would be to do experiments through SLAM, which is quite
definitely some limitations there. So this is more of a clarifying request. So I know you'd shown the beautiful difference between the measles virus and the stealth, and the difference in the neutralizing antibody responses. So my question is, for the neutralizing antibodies, given it's a pool of antibodies, do they tend to concentrate in specific parts of the HA or the F protein? So do they tend to go after, say, a more conserved region or less conserved region? And how, because the stealth is, I guess I need to rephrase that. So basically, with the difference between stealth and measles virus is there's, definitely some amino acid difference, but these neutralizing antibodies, do they tend to have an epitope which is kind of a little bit, do they tend to all go for the same epitope? Is kind of where I'm trying to ask that question. Does that make sense?
Yes, that's a great question. So we have a manuscript under review, and we saw that So we have a previous version of the virus, and we have shown that there are like 8 antigenic sites on the hemagglutinin protein, and we really need to eliminate at least 5 of them simultaneously. Okay. So if you don't eliminate 5 of them simultaneously, we couldn't see a reduction in the neutralization sensitivity. So the immune response is quite different in the case of Measles virus from influenza. In influenza, it's really focused to a few antigenic sites, and that's why the virus can escape that easily. And that was something that we have been trying to ask and to answer for so long, why it doesn't happen with the Measles virus. And we think that that's the answer. Like, well, in the case of Measles virus, the immune response is really broad. So it doesn't focus on one antigenic site. So the virus needs to mutate them all at the same time.
Got it.
And in relation with the pool, so that is PALIS. We use this pool human serum, and we have seen that this pool human serum, it has a high titer of neutralizing antibodies, not against measles, but against any other pathogens. We have seen that it targets both H and F. So we have produced like chimeric viruses in which we have measles virus H, but then we have canine distemper virus F. We couldn't see a reduction in the neutralization activity. And we did the reverse chimeric virus, in which we had measles virus F and CDV H. And the same, we couldn't see a decrease in the neutralizing titer. So that showed that, pool human sera or convalescent anti-measles virus serum target both the measles virus H and the measles virus F.
Okay.
And that's why we need to replace both proteins in the envelope.
Right, that makes sense. That makes sense. Thank you, Dr. Munoz-Alia, for a great presentation. If you'd like to learn more about the research going on at the Mayo Clinic, visit mayo.edu/research. Thank you.